All right, so lots to cover today. We'll talk about the 3 fluid compartments of the body, which should be a review. We'll talk about solute concentration and osmolality of each compartment. We've started talking about this, but we're going to add on to it. We'll talk about osmotic effects of electrolytes versus non-electrolytes. Describe factors that determine fluid shifts in our bodies, routes by which water enters and leaves the body. Describe the feedback mechanism that regulate water intake and hormonal controls of water output in urine, which is ADH and aldosterone, which you're all familiar with, and we'll talk about that renin-angiotensin-aldosterone pathway that you all should be familiar with as well. We'll talk about the importance of obligatory water losses, describe possible causes and consequences of dehydration and hypotonic hydration, which is essentially overhydration. We'll talk about the roots of electrolyte entry and loss from the body. Talk about the importance of sodium in body fluids, body fluid and electrolyte balance, so more aldosterone. Describe the mechanisms involving sodium balance, blood volume, and blood pressure, which is the RAA pathway, and then how potassium, calcium, and ion balances in plasma are regulated. So a lot of review today. So body fluids consist of water and solutes in 3 main compartments, which you should all be familiar with. So our total body water is going to be making up about 60% of our body weight, but that value, like many of the values that you will see in an average biology textbook, this is a value for the average healthy adult male. And so you should already be familiar with our intracellular fluid. That's 40% of that 60% is the fluid inside of our cells, and we have like 1 trillion cells, so lots of fluid can be held within those cells. Then the extracellular fluid is the other 20%, and that's subdivided into our interstitial fluid, which is a fluid in between cells, as well as the fluid inside of our blood, the blood plasma. So all of what I'm about to say is already in your lecture notes, but body water content is going to depend on your age, your body mass, your percent body fat, and your sex. And so infants, because they're made up of a lot of cartilage and not a whole lot of bone just yet, they're about 73% or 75% water. So a lot more water content you would see in a baby versus an adult. Like we said, a young healthy adult male would be 60% water, and then young females would be about 50% water. And the reason for that is that fat is the least hydrated tissue. So fat is only made up of about 20% water, while skeletal muscle mass is like 70% water. And males in general, like we've mentioned before, have more skeletal muscle mass. Females tend to have more fat. So because muscle mass is more water heavy, males tend to have more water in general than females do. And this is average numbers. Not, obviously if you're workout, if you train, obviously you'll have more water because you'll have more skeletal muscle mass. But if you compare the average male to the average female, the male will have more water because more muscle, and females will have less water because they have more fat than muscle. Okay, so the more muscle you have, the more water you're going to be carrying around. Any questions on that? Okay. So you should already be familiar with this image here. For sodium, our intracellular concentration is 12. And what is the units we use for ions? They use milliequivalents per liter. So these should all be milliequivalents per liter. I'm not going to write all of them out, but each one of these is milliequivalents per liter. So 12 inside the cell for sodium, 140 outside the cell for sodium. And for the most part, these 2 subsections are extracellular fluid. Their concentrations are going to be pretty equal. So they tend to be pretty much the same between the 2, except for one thing. What is one thing that we said we should not find in the interstitial fluid that we find in the plasma? Protein. So that's the one thing that they're going to differ in. Okay. So sodium is our most important extracellular cation, so our negative ion that's outside the cell. That's the most important one, while potassium is our most important intracellular cation. So for potassium, it's 140 milliequivalents inside the cell and about 4 in the extracellular fluid, both in the interstitial fluid and the blood plasma. And if you remember from B32, this is all to set up for an action potential. We have to have a lot of sodium on the outside of the cell to allow for depolarization and that sodium to rush into the cell so that we can reach that positive 30 of our action potential. And then we need lots of potassium inside the cell to have repolarization, which is the efflux, or the potassium going out of the cell. So this all sets us up to be able to communicate with our nervous system. So one thing, a clinical application is something called third spacing. And this should already be in your lecture notes. But third spacing is when we have fluid that escapes to usually a body cavity, but pretty much it's not held with any one of these three spaces. So we have fluid that accumulates somewhere outside of these three spaces. Okay? So it's abnormal accumulation of fluid. And this would be usually in a body cavity. And we would see third spacing in people who experience, let's go back. Did you guys write that down, abnormal accumulation of fluid? Hopefully you did. So third spacing, abnormal accumulation of fluid, usually in a body cavity. But you'll see this in people with burns. There's lots of fluid loss with burns. And I hate red, so I'm not going to use red. Also, edema. We've talked about edema when it comes to congestive heart failure. We've talked about lymphedema when there's maybe tumors or blockages around lymphatic capillaries. What is the edema that's specific to cirrhosis that we've talked about? And it's abdominal accumulation of fluid. Ascites. So ascites would be another example. And then people also experience this kind of fluid loss post-surgery or when they've experienced trauma. And so what makes this dangerous, it's third spacing, is because if a fluid is in a body cavity and not held within one of these three compartments, it can't do anything for the body. It can't carry out its normal functions. Fluid cannot if it's outside of these three compartments. Okay, so we have this fluid that's filling up spaces and not really doing anything for us.
What about a pregnant patient who has toxemia?
I mean, that would also be an example, yeah. Any abnormal accumulation of fluid outside of these three compartments. Okay, questions about third spacing? Okay. So we're going to do a solutions review, match the term to its definition. Our terms are solute, solvent, and solution. A is a homogeneous mixture of two or more components. B is the dissolving agent. And then C is the substance that is dissolved. So solute, would that be A, B, or C? C. It is a substance that we dissolve in a solution. What would be solvent? B, it's the dissolving agent or medium, and it's usually going to be water. And so solution is the homogeneous mixture of those two things. So a solute plus a solvent makes a solution. And that should be reviewed to you all. See, this one's not so bad. It's not a lot of review. A lot of things you should already know. Okay, everyone good here? Can I move on from this slide? Okay. So electrolytes. And this should be in your notes as well. The electrolytes are chemicals that separate electrically in fluid or solution. So any chemical that separates in solution. It could be salts, which like sodium chloride will separate into a sodium ion and a chloride ion. That would be considered an electrolyte. Acids, we talk about the bicarbonate buffer equation all the time about the splitting of carbonic acid into a hydrogen ion and a bicarbonate ion. That would be considered an electrolyte. But if hydrogen is one of these separated ions, separated ions, it's considered an acid. And you should know the difference between a strong acid and a weak acid. So a strong acid is an acid that completely dissociates in solution. It gives away all of its hydrogen ions. So hydrochloric acid would be considered a strong acid because it's made up of hydrogen and chloride, and it completely separates when it's in solution. It's a hydrogen ion and a chloride ion. That is a strong acid. Carbonic acid, however, is a weak acid because it has two hydrogens, but when it splits into one hydrogen and then a bicarbonate ion that keeps one of those hydrogens. So it doesn't completely dissociate. It does not give away all of the hydrogens that it has to give. Okay? So because it only gives one of the two that it has, it's considered a weak acid. So it's a weak acid because it releases only one of its two hydrogen ions. Does that make sense? Okay. So any questions about weak acids versus strong acids? And this is not a chemistry class, so we will only use examples that we've used before, like these two. These are the ones that you're probably going to see. Okay. I wouldn't expect you to know how any other acids separate. Okay.
Like hydrolysis is like leading that way, or
What, glycolysis? No.
hydrolysis?
Oh, hydrolysis. I mean, this would not be considered hydrolysis. Hydrolysis is when we break down polymers into monomers, which is like a covalent bond. No, this is an ionic bond where they automatically split. You don't need a process to split them. You just put them in fluid and they split all by themselves. So this would not include hydrolysis. We talk more about hydrolysis with nutrition and digestion when we actually have polymers that are held together by covalent bonds, not ionic bonds. All right, non-electrolytes are those things, those chemicals that do not split in water, that do not split in solution, do not separate. Whatever it is, it stays that chemical form. So an example is sugar, C6H12O6. You put that in water and mix it up, make sugar water, and it's still C6H12O6. It does not change. Nothing separates, nothing comes apart. Those are non-electrolytes, those things that do not split or separate in solution. Other examples of that we've used in class are lipids. Lipids also do not dissociate in solution. You would need an enzyme to break apart lipids to make them more simple. We don't have that here. We're just talking about mixing these things in water. And as you know, lipids don't really mix with water. They may temporarily, but then they're going to separate. Also, creatinine we've talked about before, which is a waste product of muscle metabolism. When we break down creatine phosphate to use some ATP, and it's a good marker to indicate CKD. Also urea, which is a product of protein breakdown. All of these stay their chemical form in solution and therefore are considered non-electrolytes. So everyone okay with that concept? Okay. So osmolality, which we've talked about before, is the number of solutes that are dissolved in 1 liter of water. You also may hear, if you read other sources, it may also say a kilogram of water, though they are the same measurement, same thing. So osmolality is going to direct the net flow of water. And so water moves according to osmotic gradients. And this may or may not be in your notes already, but water is going to move from a lesser osmolality to a greater osmolality. So basically, water is going to move from areas that are less concentrated to areas that are more concentrated in order to dilute that more concentrated area to try to make things equal. One thing about osmolality across those 3 fluid compartments is those 3 fluid compartments are going to have different makeups of solutes. So we said inside the cell, potassium is 140 and sodium is 12, but outside the cell, sodium is 140 and potassium is 4 milliequivalents per liter. So they're going to have different compositions of solutes in each one of those three compartments, but they're all going to equal, if you add up all their solutes together, each fluid compartment equals 300 milliosmoles. That is the total concentration of all three compartments. So 300 milliosmoles in the intracellular fluid, 300 milliosmoles in the interstitial fluid, 300 milliosmoles in the blood plasma. So osmotic power as far as electrolytes versus non-electrolytes go. Electrolytes, or actually we'll say non-electrolytes first. Non-electrolytes contribute 1 solute to solution. Because remember, non-electrolytes stay in their form. So C6H12O6 stays C6H12O6. And so it only contributes 1 solute, 1 molecule of glucose. However, electrolytes split. They split in water or solution. And so electrolytes, they will contribute 2 or more, depending on the chemical, solutes. So sodium chloride would contribute 2. Hydrochloric acid would contribute 2. Carbonic acid would contribute 2. So which of these would have a greater ability to cause fluid shifts? Electrolytes or non-electrolytes? Electrolytes, right? That's going to be, you can have more solutes on the side that has more electrolytes because they split. Which is why you drink Gatorade if you want to get hydrated. You get more electrolytes in your body, and water is going to want to follow wherever those electrolytes go. So for these electrolytes, we use milliequivalents to measure them because it takes their electrical charge into account. Are they a plus, 2 plus, minus, 2 minus? Milliequivalents takes that into account. Non-electrolytes. And I think this is already in your notes, as it shows the measurements for non-electrolytes. Yeah, so non-electrolytes are measured in milligrams per deciliter, or the same thing would be milligrams per 100 milliliters. Because we don't have to take electrical charge into account because they don't split into ions. They're not held by ionic bonds. Okay, questions about osmolality? Yeah, Isaac. What was that? Oh, they're measured in milligrams per deciliter because they're not made from ionic bonds. So we don't have to take charge into account, which is why we don't use milliequivalents. Milliequivalents are only for electrolytes that have ionic bonds, that have electrically charged ions that are bonded together. So because non-electrolytes don't have ionic bonds, we use a different measurement, milligrams per deciliter. Was that your question, Isaac?
No, I was going to say, because obviously, like you mentioned Gatorade.
Uh-huh.
Is there like a maximum limit of those types of ions that we can take per day?
Well, remember with our urinary system, there's a transport maximum for each one of those solutes ions that we're reabsorbing. So there will be a max, and whatever else is left over will just end up in our urine. Yeah. All right, so, and then we'll talk about why, like if you have too much sodium, if you have too much of these ions, we'll talk about what happens. Okay. If you take a look at this chart, you'll notice, like I said before, the extracellular fluid, both the blood plasma and the interstitial fluid, are pretty similar in their concentrations. So for sodium, the plasma and interstitial fluid are pretty similar. Potassium is pretty similar. Calcium as well, magnesium, bicarbonate, chloride, hydrogen phosphate. What is this? Sulfate. Sorry, I'm not a chemist. And so the extracellular fluid is pretty similar, or pretty much equal to each other, except when it comes to protein. So you'll see protein, lots of protein, well, a good amount of protein in the blood plasma, even more inside the cell, but you'll see little to none in that interstitial fluid. So if you look at your lecture notes, there's a table that tells you all the different concentrations of the solutes that are important to the human body. So I know we learned about sodium and potassium already before, but now make sure that you pay attention to the levels of calcium, the notes about chloride, You already know where proteins should be and where they should not, and then also bicarbonate. I don't know if this was fixed yet, but the third column on this chart here, they all should say 300 milliosmoles. If there's like a typo at the very bottom of one, just fix it. It should say 300 milliosmoles for all three. Does it? Did I fix that already? Okay,
Yeah.
good. Okay. All right. So we just went over that already. All right, so here's a review question. You're going to tell me if each one of these items here are a chemical, if they're an electrolyte, or if they're an acid. So glucose, C6H12O6, is it a chemical?
No.
Yes, anything that's made up of elements is a chemical. Okay. But is it an electrolyte? No, it does not dissociate in fluid. So is it an acid? No. Carbonic acid, is it a chemical? Yes. Is it an electrolyte? Yes. Is it an acid? It's in the name, carbonic acid. But it's a weak acid, okay, because it only gives away one of its two hydrogens. HCl, is it a chemical? Yes. Is it an electrolyte? Yes, it does split in solution. Is it an acid? Yes, it's a strong acid. Sodium chloride, is it a chemical? Yes. Is it an electrolyte?
Yes.
Yes, it does split in water, but is it an acid? No, it does not have a hydrogen to give, so it's not considered an acid. It's a salt. Okay. So make sure you know all acids are electrolytes, but not all electrolytes are acids. Okay. Last one is urea. Is it a chemical? Yes, it's still a chemical. Is it an electrolyte? No, I said it does not dissociate in solution. So is it an acid? No. If it's not an electrolyte, it's not an acid. It doesn't split. Anything that does not dissociate in water is not an electrolyte.
Professor, can you repeat, you said all electrolytes are,
All acids are electrolytes, but not all electrolytes are acids. Because electrolytes are anything that dissociates chemically in solution, but in order to be an acid, one of those things that separates, one of those ions that separate have to be a hydrogen ion. And that's not true for all electrolytes. So all acids are electrolytes, but not all electrolytes are acids. Okay, everyone good here? Okay. So fluid movement among compartments. So anytime we bring in something new, whether it's the oxygen we bring in through the lungs or the nutrients we bring in through our digestive tract, it has to travel through the blood plasma and then through the interstitial fluid in order to get to the cells that use those products. Okay. So just a review, what is the barrier between the blood plasma and the interstitial fluid? It's not a plasma membrane. What is the wall that separates the blood from the interstitial fluid? Capillary wall. It is the capillary wall. You want to say capillary because it really is at the capillary where things are being exchanged. It's not at arteries, it's not at veins, it's at capillaries where exchange happens. Okay. So then now what would be the barrier between the interstitial fluid and the intracellular fluid? That would be the plasma membrane. And then make sure that you remember the forces that are going to be shifting fluid and solutes in one way or another. We have, what does HP stand for? Hydrostatic pressure, that is a pushing force, usually caused by blood pressure. And then what does OP stand for? Osmotic pressure, that's more of a pulling force, that is the attraction of water towards one area or another. Okay, any questions about that? Okay, so we're gonna do a little bit of review on water movement. What is the transport mechanism that moves water? Bio 32. Osmosis. So we're going to do a little review on osmosis. Okay, so what is the term that we give when there is a lower concentration in the fluid outside the cell compared to the inside the cell? Hypotonic, yeah. If there's a lower concentration of solute outside the cell, it's considered hypotonic. That cell's in a hypotonic solution. What is the term we use if there's an equal concentration of solutes outside versus inside the cell? Isotonic. And then what if there is a higher concentration of solute outside the cell? Hypertonic. With a hypotonic solution, which way does water tend to move? Into the cell or out of the cell? Into the cell, yeah. So if what color do I want to use? Okay. Let's say outside the cell, we had like 2% sodium chloride. Inside the cell, and I'm just throwing out random numbers. Inside the cell, we had 4%. So it's less concentrated on the outside. And like you guys said, water is going to tend to move into the cell in order to bring this concentration down. And by removing water from that outside environment, that's going to bring this concentration up until they reach in equilibrium. Okay. Everybody's still okay with that from last semester? Okay. Isotonic solution. Which way is water going to move? Into the cell, out of the cell, neither, both? Which one? Not neither. It's always going to be both because water never, ever stops moving. You're never going to get water to just not go anywhere, even in an isotonic solution. So that just means that the rate that the water is moving into the cell is the same rate that it's moving out of the cell, so things stay equal. Okay, so don't ever say neither. It's both. Okay, moving in both directions because water never stops moving. It's always moving. And then I forgot to mention this, but with that hypotonic solution and water moving into the cell, that cell is going to swell. And if the gradient is too different, if there's too big of a difference in concentration and too much water moves into that cell can lyse and burst. We had talked about like water toxicity, and ingesting too much water could make your cells burst because we only have a plasma membrane and it can only withstand so much pressure. Like plants, on the other hand, they have a whole cell wall that makes their membrane like stronger, and they can hold extra water to stand up, but that's not the case for humans. We can only withstand so much pressure before those cells burst. And so with an isotonic solution, would that cell get bigger, smaller, or would it stay the same? Stay the same. Okay. If we're talking about a red blood cell, it's going to maintain that biconcave shape. And then a hypertonic solution, which way is water going to want to move? Out of the cell. And then what's going to happen to that cell? It's going to shrivel, or you can say shrink, you can say crenate. All of those terms mean the same thing. But water is going to go out of the cell to try to dilute that higher concentration outside the cell. So everyone okay with that? And the term I always use is hypo makes a hippo. And a hypotonic solution is going to make the cell swell and get bigger. So hypo-hippo. Okay. This is just another diagram showing you what happens in these different solutions. So again, isotonic, the cell is going to stay the same because water is moving in and out at the same rate. Hypertonic solution, that cell is going to shrivel or crenate. And then with a hypotonic solution, that cell is going to swell. And either one of these two over here, the crenation or the swelling, that is going to mess with cell function. If there's too much water or not enough water, then you're going to have some dysfunction in those cells. Questions there? Okay. So the next key concept is that both intake and output of water is regulated. So these little vials here are showing you the average intake per day and the average output per day. So the water that we intake, 60% of that comes from the beverages that we drink, 30% of that comes from the foods that we eat, and 10% of that is from metabolism, and we call that metabolic water. So there are some metabolic processes that do make water, like dehydration. You remove a water molecule. At the very end of anaerobic or aerobic respiration, some water is made as well. And so there are metabolic processes that happen that create water, but that accounts for very little of the water that we make. One thing to remember, and you can see both of these vials are pretty much equal. So your input of water and your output of water is equal each day, or pretty much equal, in order for you to stay hydrated. Okay, which is about 2,500 milliliters. Which equates to, because we're in the U.S., it's about 84 and a half ounces. Our output of water, 60% is from urine, 8% from sweat, 4% from feces, and these three here are what we call sensible losses. Sensible losses are those things that can be measured. Measurable losses. We can measure the amount of urine output in a clinical setting. We can measure the amount of sweat. It's a little bit harder, but you still can measure sweat. Feces is measurable as well. Insensible loss are those things that you can't measure. So the water that evaporates from your skin when you're hot, we can't measure that. The water that escapes from your lungs when you breathe out, we cannot measure that. And so it's considered an insensible loss, but it does account for about 28% of the water that we lose. And so either a decrease or increase in osmolality or concentration will trigger a homeostatic feedback mechanism. Can I erase this little thing that says sensible losses? Okay. So if we experience an increase in osmolality, where our body ends up being more concentrated than it should be, that's going to trigger our thirst mechanism so that we intake more water, dilute the concentration that's within our body to bring us back to normal. And what is the hormone that's going to cause us to reabsorb more water? ADH. And so we'll also have ADH release, which is going to allow us to reabsorb more water, again, to dilute that high concentration. What are the proteins that are responsible for reabsorbing water when we have ADH release? Aquaporins. One more thing before you take a picture, Molly. If we have a decrease in osmolality, all that's going to happen is we're going to inhibit those two processes. So if we have a decrease in osmolality, our body is already diluted in concentration, then we're going to inhibit that thirst mechanism so you're not drinking any more water, and we're going to inhibit ADH release so you're not reabsorbing more water into the blood. And then also remember that sodium and water are very closely tied. Anywhere sodium goes, water is going to follow. All right, everyone okay here? All right. So your thirst mechanism. What kind of receptors detect change in osmolality? Those are osmoreceptors. How they detect osmolality is they're actually measuring plasma membrane stretch. So they're really looking out for a loss or gain of water. Have we gained water? Has that cell stretched? Are we losing water? Has that cell shriveled or crenated? And we only need an increase of about 1 to 2% in osmolality to trigger these osmoreceptors. So it doesn't take much change for homeostasis to come in. So again, if we have an increase in osmolality, like we just said before, it's going to trigger our thirst mechanism. And so what we'll end up seeing is we'll have dry mouth. What is the term for dry mouth that we learned about before with digestive? Halitosis is the bad breath that you get from dry mouth. Xerostomia. And so we get xerostomia because our hypothalamus is reducing the work of our salivary glands. So salivary gland secretion is reduced. And again, this thirst mechanism comes from the hypothalamus. So another way that the thirst mechanism is triggered is also if we experience a decrease in blood volume. So if our blood volume goes down, we don't have enough water in our blood, that's also going to trigger the thirst mechanism, give us dry mouth so that we bring in more water and increase our blood volume. So if you experience a decrease in 5 to 10% of blood volume, that will trigger this thirst mechanism. And that, like in the case of a hemorrhage. So if you lose lots of blood volume goes down, you're going to feel pretty thirsty, so you intake more water and bring that blood volume back up. What are the, because blood volume is directly related to blood pressure, what are the receptors that detect changes in pressure? A very specific type of mechanoreceptor. Baroreceptors. These changes in pressure. And so if we have a drop in blood volume and blood pressure, that's going to trigger that RAA pathway that we talked about. And what is the term for excessive thirst in patients? Polydipsia. Anybody still need these notes? Okay, go ahead. Good? Okay. So this is just a diagram showing everything we just talked about. If your body is too concentrated, you experience an increase in ECF osmolality. So for example, your plasma concentration goes up. You're going to have a decrease in saliva and that dry mouth. You have osmoreceptors that detect that in the hypothalamus, which is going to trigger that thirst center. You're going to have a sensation of thirst. So hopefully you drink some water. The water moistens the mouth and the throat and stretches the stomach and intestines, and that extra water you just ingested is going to be absorbed from the GI tract, which should decrease your osmolality back to normal. Okay. You'll see that same mechanism happen in a different way, though, if you lose plasma volume. So if you do have hemorrhaging or excessive vomiting or diarrhea where you're losing lots and lots of water, this is going to decrease your blood pressure, which causes the granular cells in your kidney to release renin. That renin converts angiotensinogen into angiotensin 1. Then we have an enzyme from the lungs that converts 1 into 2. And then angiotensin 2, one of the 4 pathways for angiotensin 2 is that thirst mechanism in the hypothalamus. So again, you bring in more water, it stretches your intestines and your stomach, you absorb more of that water through your GI tract, and now your plasma volume is back to normal. So everyone okay with that concept? Okay, any questions about that? Okay. So obligatory water losses. So we already talked about those obligatory water losses over here. We have to lose water through our feces, sweat, urine, and then also from our skin and lungs. Beyond that, though, and this might already be in your notes, but beyond that, output depends on intake. So if you're bringing in more water than that 2,500 milliliters we just talked about, then you're going to be outputting more water. If your diet has more water content than the average person, then you're going to be outputting more water. If you have a diet that's high in vegetables, vegetables have a lot of water content, then you're going to be outputting more water. Also, water loss. So what I mean by that is if you increase the amount of water you lose by sweat, then that's going to lead to you urinating less. So the more water you put out in one mechanism, then you're going to have less of it in another. When you do intake excess water, there's about a 30-minute delay before we inhibit that ADH, before we kind of slow down the water reabsorption because you've taken in too much water. Okay, so there's about a 30-minute delay. So when you drink that bunch of water, it'll take about 30 minutes before you start seeing an increase in your urine output. So this is the same thing here we've talked about. If you experience an increase in osmolality or a decrease in plasma volume, this is a different mechanism that helps us replenish that water. So we have osmoreceptors in your hypothalamus and baroreceptors in your atria and your carotids and your aorta that are going to detect these pressure changes. So if you increase concentration or if you lose plasma volume, that is going to cause your posterior pituitary to release more ADH, which is going to put more aquaporins in the collecting duct, which causes more water reabsorption, which should decrease your osmolality or increase your plasma volume. Well, it'll do both, but depends on what was the trigger that started this whole thing. And because you're reabsorbing more water, your urine output will be lower. See, so much review. And that is all part of this RAA pathway. If you experience a decrease in arterial pressure, it's going to inhibit or trigger your baroreceptors to increase your sympathetic nervous system. We have release of renin, which is going to convert angiotensinogen into angiotensin 1. Then we have this angiotensin converting enzyme, which organ did I say it comes from? The lungs. That's going to convert angiotensin 1 into angiotensin 2. We just talked about the thirst mechanism and ADH release, but we'll also have some aldosterone release from the adrenal cortex so that we reabsorb sodium, and sodium and water are closely tied. So anytime we reabsorb sodium, we'll also reabsorb water, which is going to increase blood volume and increase blood pressure. And then remember, through this pathway, we'll also get vasoconstriction to have a temporary increase in blood pressure. But remember, this is a short-term response to help with that blood pressure issue on the short term. But anything that changes blood volume, so these three pathways here, because we're dealing with kidney function, which takes a little bit longer, but also lasts longer, this is a long-term response. So we have a way of dealing with the blood pressure issue quickly, and then also for the long term. So you can guess, there's going to be a good amount of questions on the RAA pathway for sure. Because we talked about it during blood, we've talked about it during urinary, we're talking about it again here. So it's a reoccurring theme, so expect to have it on your final exam. Disorders of water balance, dehydration, and hypotonic hydration. We'll talk about both of those things. So dehydration, here we have the girl running in the desert again. So if you're running in a very hot environment and sweating a lot, what's going to happen is you're losing lots of water from your extracellular fluid because of the sweat that you're experiencing. And then what's going to happen is that the concentration inside your ECF is going to increase. You've just lost water, so it's going to make it more concentrated here. And so now your cells are in a hypertonic solution, very concentrated on the outside of the cell, and that's going to cause water to move outward. And that's going to cause your cells to shrivel because you've lost so much water. It causes this big fluid shift to the outside. And so you're going to see this kind of movement, not only with excessive sweat, but you'll see this also with hemorrhaging, diarrhea. What is the word for vomiting that we learned? Emesis. If you abuse diuretics, this will be a consequence as well. Or if you have a very, very, very low intake of water, if you're not really drinking any water at all, you'll experience dehydration. Now and you're going to experience symptoms that are similar to a high volume blood loss that we talked about during the blood chapter. You guys remember what that's called?
What was the question again?
What was the clinical from blood where you have a high volume blood loss? Hypovolemic shock. So these people with severe dehydration will experience the same symptoms as that of hypovolemic shock. All right, on the other side, hypotonic hydration is essentially overhydration. And so again, this girl who just finished off 5 cups of water and she's having another one. What's going to happen is excessive amounts of water are going to enter the ECF. And this can happen because, remember, it takes about 30 minutes for us to have an inhibition of that ADH. So if you drink a lot of water in a short amount of time, your body's going to absorb that. Okay, so you have water moving into your extracellular fluid, which is going to decrease osmolality. So you're going to have less of a concentration on the outside of the cells because you just diluted it all by bringing in too much water. So now your cells are in a hypotonic solution. Hypo makes a hippo. Water is going to want to go towards where it's more concentrated. And so these cells are then going to swell and could eventually burst depending on how much water you actually intook. Now, the biggest issue that occurs with hypotonic hydration, what's most dangerous, is that this will cause hyponatremia. And hyponatremia is low sodium levels in the blood. Sodium is so important for cell-to-cell communication, nervous system activity. We need sodium. And so if we overhydrate and experience hypotonic hydration, we're going to end up with our cells not having enough access to that sodium. And so this leads to nausea, vomiting, muscle cramps because you're messing with membrane potentials, active potential to move, and it can actually lead to cerebral edema, swelling in the brain.
It passed away from the water challenge.
Yeah. It was Don't Go Wee Wee for a Wii. It was to win a stupid Nintendo Wii and she died.
Yeah.
And the cerebral edema can lead to disorientation, convulsions, coma, and unfortunately also death. Treatment would be a hypertonic saline solution. So we'll just pump them up with a bunch of sodium because they're lacking it. Well, if you drink a lot of, or intake a lot of salt, that would be hypernatremia, and we'll talk about that next.
You gotta do it at the same time.
Water and salt at the same time. All right, can I erase the hyponatremia notes yet?
What is that
Yeah, treated with hypertonic saline. So a highly concentrated sodium fluid. So everyone okay with those hyponatremia notes? Okay. So the last disorder of water balance, which you should already be familiar with, is edema. Ooh. Which is when we're talking about just edema and not third spacing or anything else, if it's just edema, we're talking about filling up a fluid in the interstitial fluid only. And this is considered tissue edema or local edema. And so what makes edema dangerous for the body is because we're increasing the distance required for oxygen and nutrients. I'll write it over here, oxygen and nutrients. Because our oxygen and nutrients need to go from the blood to the cells. But if our interstitial fluid, that middle compartment, is filled with a lot of fluid, a lot of water, then now that oxygen and nutrients has a longer distance travel to get to the cell. So then we get a lower amount of oxygen delivery, a lower amount of nutrient delivery, which is going to cause function to go down if we're lacking oxygen and nutrients. Lack of energy, lack of the substances that are needed to build proteins and such in the body. So it just makes it a lot harder on the body if you have edema. It's a longer way to travel. Okay. Any questions there? Okay. Next, we're going to talk about sodium, potassium, calcium, and phosphate levels and how they're tightly regulated. So we'll talk about salts first, which salts are electrolytes. Dietary intake or the entry of salts include food. We have salt that we get from food, fluids, and some of the salt we get is metabolic, and that is from breaking down bone via osteoclasts. So when an osteoclast break down bone, not only do they release calcium, but they release some salts as well, phosphate salts that are in our bone. How we get rid of salts is through our sweat. Sweat is normally hypotonic, so not very concentrated. But the more water we lose through sweat, the more salt we're going to lose as well. We can lose salt through our feces. There are certain GI disorders that we won't get into, but there are certain GI disorders that can increase salt loss, where it doesn't allow us, these GI disorders don't allow us to absorb the salts, and so we just lose them in our feces. We'll also lose some salts through our urine as well as our vomit.
So if we have an increase in parathyroid hormone because the osteoclast activity is increased, we're gonna have more salt.
Yeah. So Caitlin just asked if you had an increase in parathyroid hormone where you're breaking down bone to release calcium, you would also be releasing those salts. So you could have a salt issue if you have overactivity of your osteoclast. Anybody still need this up? Okay. Addison's disease. I think we talked about this with endocrine. You guys remember Addison's disease? Yeah. So it's a lack of aldosterone. Where does aldosterone come from? Where is it released by?
The medulla, adrenal?
Adrenal what?
Medulla?
Cortex, the adrenal cortex. Another hormone that's released by the adrenal cortex, anybody know? Besides androgens, what is the other hormone released by the adrenal cortex? Cortisol. So usually if you lack aldosterone, you'll lack cortisol as well. So those usually go hand in hand. But when you lack aldosterone, you're not reabsorbing that sodium, and so sodium leaves by the urine because it's not being reabsorbed, and water is going to follow wherever that sodium goes. So if more sodium is left in the filtrate in urine, water is also going to be left in that filtrate in urine, and you're going to end up with more urine output. And so because sodium leaves by your urine, and you're lacking that sodium, lack of sodium can cause extreme fatigue. Sodium is so important for nervous system function. You may experience weight loss because the amount of water that you're losing. Because you're losing so much water, you'll have a decrease in blood pressure, which can lead to fainting. And then having low nervous system function because of the low amount of sodium, that can also lead to depression. And what ends up happening with people with Addison's disease is they'll crave salty foods. Oh, because of the lack of nervous system function. So sodium is really important in nervous system function. If that depresses, it's going to also depress your mood and everything else. And two, fatigue. Being extremely fatigued can cause depression as well. They're all kind of related to that nervous system function. All right, pica is another disorder involving salt or electrolyte, more specifically. It's usually caused by a lack of minerals. The lack of minerals, the usual culprit is usually iron. You're lacking iron, but it can be a number of other minerals. It makes you crave abnormal substances. Does anybody still need those? No. Okay. It makes you crave abnormal substances like chalk, like clay, like burnt match tips. Also could be dried paint. So because you lack this mineral, your body's going to crave wherever it believes this mineral to be. Could be dirt as well, or soil. Okay. And so how you do this, how you treat pica, people who are eating these abnormal substances due to a lack of minerals, and this is more common in people with pregnancy. So pregnant people can experience this as well. But what you want to do is first you want to treat any complications from eating these non-food items.
Oh, like people actually.
Yeah, they actually will eat it. They will ingest these things. So you treat the complications first, while also treating the imbalance. You want to make sure that you're treating any kind of complications that come from eating the chalk, clay, or burnt match tips, or soil, or whatever it is that you're eating that you're not supposed to be eating, but then also take care of the imbalance. What is that person lacking? Give them what they're lacking, whether it's iron, calcium, whatever it is, what mineral they're lacking.
Ice chips too, crushed ice.
Yeah, that could be one of them as well. Yeah. I have a really weird food craving. It's so weird. When I was little, this is so weird to admit. When I was little, I used to like the carpet fresh that you would sprinkle on the carpet before you vacuum, and I would like taste it. What's weird is that now if I smell it, my mouth waters. Like I don't eat it anymore. I'm not 5 years old, but it still makes me salivate when I smell it. It's so weird. Brains are weird.
Do you remember the paste in kindergarten?
Oh, yeah.
I remember little friends, they used to like
Eat the paste. Yeah. Kids are weird. I was weird. What were you were going to say something, Caitlin?
She mentioned ice. I'm anemic and I
Oh.
only crave ice all day long.
Yeah,
Like, I
yeah.
can sit and chew on ice.
Yeah, that's a common thing with people who are anemic. They still want to chew on ice. You what?
I got an ice maker.
Oh, did you?
Sonic ice.
Oh, yeah, my mom got me one for Christmas. Oh, I love it so much. All right, so we talked about how sodium is so important. It's so important that it's 90% of the solutes that we see in the extracellular fluid. It's 90% sodium. So out of those 300 milliosmoles, 280 of that is from sodium. We get sodium from sodium bicarbonate. We get sodium from sodium chloride. Those are the main sources of sodium for us. Hypernatremia. So what did we say was the average value for sodium outside the cell? 140. So if it's greater than 145 milliequivalents per liter in the blood, it's considered hypernatremia. If it's lower than 135 milliequivalents per liter in the blood, it's considered hyponatremia. Both of these are going to affect water balance. So we already talked about hyponatremia. Right now we're going to talk about hypernatremia. Hypernatremia is due to either inadequate water intake, fluid loss, or in very rare cases, an extreme increase in sodium intake. But this is rare, like if you were dared to do so. So symptoms of hypernatremia are going to be, and some of these can be considered signs as well, is going to be fatigue. You're going to have fatigue anytime you have an imbalance in sodium. Okay? So fatigue, excessive thirst. Ooh, that is not how you spell excessive. And again, what's the term for excessive thirst? Polydipsia. Excessive thirst, muscle twitches, and also seizures or convulsions. Oops, seizures. And then you would treat with water intake, or IV fluids. Those patients, so some patients that have problems with too much sodium or hypernatremia, they can actually, people who have troubles with blood pressure as well, because remember sodium, water follows sodium. They sometimes will use salt substitutes, so they won't use sodium chloride for salt if they have problems with sodium. They'll have maybe potassium chloride instead, which is still, you can still use it as salt, it's the same thing. So salt substitute, I'll write that down, salt substitute. And I think it's already later on in your lecture notes, so we'll talk about it again. So those with issues with either sodium or issues with blood pressure can have salt substitutes like sodium chloride. But if you're going to use sodium chloride as a salt substitute, your aldosterone has to be functional. Remember, when aldosterone is working to reabsorb sodium, it's secreting potassium in the other direction, because our blood levels of potassium need to be really low. So it needs to be 4 milliequivalents per liter. So we need to be able to get rid of any excess potassium. So if you're using a salt substitute, you have to have good functioning kidneys and good functioning aldosterone in order to make sure that we're not having too much potassium. We need to have a mechanism to get rid of that potassium. You were going to say something, Caitlin?
My sister has POTS, and she had hypernatremia, and her hands literally got stuck, stuck in a contracted way. And they had to give her 4 bags by fluids. It was really bad.
Yeah,
And
yeah.
literally she couldn't move her hands.
Yeah, sodium is so important with nervous system function. Yeah.
All of the signs and symptoms and symptoms is all from both hypernatremia and hypo.
No, just hyper. We already talked about hypo. This is all symptoms for hypernatremia.
Professor, the salt, potassium chloride, that's one of the treatments.
That if somebody has an issue with hypernatremia or blood pressure, instead of taking more sodium chloride, more of that kind of salt, they can use a salt substitute instead. So they're not ingesting more sodium.
random question. Does the salt substitute also have iodine?
Oh, is salt substitute iodized? I don't know. Hey Siri. Hey, oh, because I have it on Do Not Disturb. Somebody else says Siri, go, huh? Hey Siri. Is potassium chloride iodized?
Potassium iodide, chemical formula key, is a salt that is similar in structure and physical character to common
Potassium,
table salt.
no, that's not the
Sodium.
answer I want. Or hey Siri. Hey Siri. Are salt substitutes iodized? Oh, it doesn't even say that. I want to know though before we move on. Because you're right, if you're using a salt substitute, then and it's not iodized, then you're going to have to look for other avenues. No, potassium chloride is not iodized. No. So then you would have to worry about making sure you get enough iodine in your diet as well. Hey, that's a good point. So here's that here, the influence of aldosterone. So again, if we have not enough sodium or if we have too much potassium, that is going to trigger the adrenal cortex to release aldosterone, which is going to target the kidney tubules to activate that sodium potassium pump. So if we're lacking sodium, we reabsorb more of that sodium, or if we have too much potassium, we're secreting that potassium out. And that should bring us back to homeostasis in regards to sodium and or potassium. Okay, what are the cells in the urinary system that detect levels of sodium in the filtrate? Say it louder. Say it with your chest. What are the cells called? Somebody said it. Macula cells. And the macula cells and the granular cells make up what structure at the nephron? The juxtaglomerular complex. Juxtaglomerular complex. You cannot say JGC. Juxtaglomerular complex. Come on. You can if you want, just half a point and not a full point. All right. Any questions? This is just a diagram that's kind of summarizing all of these pathways that we just talked about. So make sure that you're able to read this, follow along, and understand the physiology that's happening here. ANP. ANP is an antagonist to which hormone? Aldosterone. So aldosterone is released part of the RAA pathway when blood pressure is too low. So when blood pressure is too high, ANP does the opposite. So you can see the trigger here is the stretch of the atria of the heart due to a high blood pressure. Because remember, ANP stands for atrial natriuretic peptide. Okay? So it's produced in the atria. So the atria experience that increase in blood pressure. We release the ANP. And the ANP is going to, the biggest thing it's going to do is inhibit aldosterone, but it's really inhibiting this entire RAA pathway. We'll have less ADH release, so less both sodium and water reabsorption. And we'll have because we have an inhibition of this pathway, instead of those vessels vasoconstricting, you're going to have vasodilation in order to bring down that blood pressure. So we say it's an antagonist to aldosterone, and it is at its most important role, but it's really inhibiting that entire RAA pathway. So less sodium reabsorption, less water reabsorption, no thirst, and vasodilation.
Professor, is that considered short-term?
The vasodilation would be short-term. Everything else would be long-term. All right, influence of other hormones. I think this might be in your lecture notes, so I don't have to write it all out. No, I'll just write it. estrogen is chemically similar to aldosterone. So estrogen, unfortunately, causes increased sodium and water retention, which is why you get, not you, everyone, but women get bloating during their cycle. And usually they're most bloated right before ovulation. Okay. And also the reason why you have water retention in pregnancy. The estrogen causes more salt retention, which causes more water retention. And if you remember from female reproduction, don't forget those hormone pathways, that because of the FSH and LH pathway, we're going to have an increase in estrogen. And then that's going to cause a negative feedback, which causes a decrease in FSH. Only one of the follicles that are ready to ovulate are going to be able to survive that drop in FSH, and that's our dominant follicle. And that's going to happen right around here. And when that dominant follicle is chosen, it produces lots and lots of estrogen. That's going to cause a surge in a hormone. What is the hormone that we experience the surge in? LH. And that LH surge is directly responsible for ovulation. How many of you have watched the female reproductive lecture already? Oh, that makes me so happy. Okay. Now, progesterone has an opposite effect. And because of progesterone, you should experience some water loss right before your period. Now, obviously, this is generally what occurs, but your diet and your activity in your life can make you bloat in other parts of your cycle due to whatever you're ingesting or doing. Anybody remember why progesterone is a good birth control method? What does it do? It makes your cervical mucus thick. So then the sperm have a hard time getting to the egg because the fluid's too thick. Okay, clinical note on Cushing's disease. We said Cushing's disease back in endocrine was one of those ones that is most, it's one of those ones that are most commonly caused by an overprescription of steroids. So when people come for like pain management and stuff, they are prescribed or given injections of glucocorticoids too often, which can lead to Cushing's disease. So it's an excess levels of steroids or cortisol, which can be doctor mediated, so you're prescribed too much of this, or if you have a chronic disorder that causes the release of cortisol on a regular basis. So things like asthma or rheumatoid arthritis that causes a lot of stress on the body and causes a lot of cortisol release can lead to Cushing's disease. Also, if you have a tumor, and then which is the gland that releases cortisol? Adrenal cortex. If you have a tumor on the adrenal gland, specifically at the cortex, it may release excess amounts of cortisol. What do we call that when we have a tumor on a gland and it retains its ability to secrete the hormone? Secreting tumor. So if you have a secreting tumor on the adrenal cortex, it can cause excess levels of cortisol. Also, if you have a pituitary tumor as well, because ACTH, which stands for adrenocorticotropic hormone, is going to cause the release of cortisol from the adrenal cortex. So if you have a tumor that releases too much ACTH, then you're going to have too much cortisol release as well.
Do you mind spelling out
Oh, rheumatoid arthritis. So one of the things we talked about with Cushing's disease is that cortisol, it causes mobilization of fat, so it makes it so that more fat is available in the blood in case the stress that you're experiencing is life-threatening. Okay, you're starving, you don't know where your next meal is going to be, so your body makes it so that more energy is available in the blood. But if your cortisol release is due to pain or due to some non-life-threatening situation where you don't use that fat for energy, then that fat gets redeposited, and it usually gets redeposited at the back of the neck, causing a buffalo hump. Huh? Amy Schumer? Is it Schumer? Yeah, Amy Schumer, yeah. Another thing that excess levels of glucocorticoids do also, we have enhanced tubular reabsorption of sodium. So we're also going to see some edema, so some abdominal swelling as well. So people with Cushing's disease, they may gain a lot of fluid in the abdomen, which is going to cause stretch marks if the fluid accumulation happens very fast. And so not only do we have fat movement because of this cortisol release, but also water retention as well. All right, questions on Cushing's?
Can that cause ascites?
I don't know if we would I don't know if we would call it ascites in this case, but I'll let you know. Yeah.
Professor, I've noticed a lot of the young kids now, like with, they're always on their phones and stuff, their hair looks like this and everything like this. Is that also considered a buffalo hump?
The buffalo hump is usually a fat pad, so if it's tension and it's more connective tissue, then it wouldn't be a buffalo hump.
This posture,
Yeah, bad posture. Anybody remember what the spinal defect is if you have an over-exaggerated thoracic curvature? Lordosis is down here. What's up here? Kyphosis. Not that I'm going to ask you that, but just I want to see if you remember. All right. So we're almost done. What time are we at? 9:21. Okay. Regulation of potassium. We said that potassium is the most important intracellular cation. So the ion we have inside of the cell. It should be about 3.5 to 5.5 milliequivalents per liter in the blood, or ECF, and I believe all of this is in your lecture notes. Yeah, it is. It's necessary for membrane potential. We talked about that. We need it for the repolarization of our cells. We talked about it during heart, that having too much or not enough potassium, hyper or hypokalemia, can cause fatal arrhythmias as well. And because aldosterone secretes so much potassium, we're more worried about reabsorbing sodium than we are keeping levels of potassium high. You need to make sure that you're intaking an adequate amount of potassium from your diet. And you can get potassium from meats, dairies, vegetables, grains. There's a lot of different sources of potassium. So you have to make sure, bananas are a big one that people say. So you need to make sure that you're taking in, enough potassium because your body secretes it so much. You have a very limited ability to retain potassium. So you need to make sure you're getting enough of it in your diet. Okay. Questions about potassium? Okay. One thing that I want to talk about, and I want to make sure we talk about this before we leave and talk more about fluid balance in lab, is that acidosis can lead to hyperkalemia. And hyperkalemia is too much potassium in the blood. So when you have acidosis or a low blood pH, now what should your blood pH be normally? 7.35 to 7.45. So if you have a low pH, what ion do you have too much of? Hydrogen ions. Hey, if you have a low pH means too many hydrogen ions. So if you have too many hydrogen ions, here's the plasma, here's a red blood cell. If you have too much of these hydrogen ions, they're going to start to diffuse into your cells because there's an excess of them and they're going to start going in places they shouldn't. Okay, so you'll start to see hydrogen ions go into the red blood cells. And we need to have electrical balance in order to maintain memory potential. So if we gain a positive, we usually need to release a positive. And so we said that our potassium is the most important intracellular cation. We have 140 milliequivalents of potassium inside our cells. So that is the most available positive ion that we have. So when we're bringing in excess hydrogen ions because of the acidosis, we end up exchanging it for potassium. And then we end up with too much potassium displaced into the plasma, causing hyperkalemia. Okay? And hyperkalemia can lead to fatal arrhythmias. So usually when somebody is experiencing acidosis, they will also experience hyperkalemia because too many hydrogen ions are going into their red blood cells, and then your red blood cells are exchanging that with potassium, putting too much potassium in the blood. Okay. It's 9:25. There's 10 more minutes until the next class comes in. So I'm going to go through the next 2 slides. You're just going to sit here and you're going to listen. Hopefully. Okay. Everybody okay with acidosis and hyperkalemia being linked? Okay. Do you want to go to the next slide, please? Okay. I'm not going to talk too much about regulation of calcium. You should already know this. Calcium, when blood calcium is too low, we call that hypocalcemia. When blood calcium is too low, it triggers the release of parathyroid hormone from the parathyroid glands. Parathyroid hormone has 3 different targets. It's going to target the osteoclast to break down bone to release more calcium into the blood, bringing it higher, closer to normal. Also going to target the kidneys to reabsorb more calcium to also bring it up. And then lastly, it's going to activate vitamin D in order to cause more absorption of calcium at the intestines. So we're going to target the intestines to absorb more calcium, our kidneys to reabsorb more calcium, and then your osteoclast to break down bone to release more calcium into the blood. But that's review. We've talked about that lots of times. Okay. Regulation of anions. So one thing about ions is that positive ions need to be balanced with negative ions a lot of the time. So one thing we haven't talked about yet, but it's in this diagram here that we saw with urinary, is that sodium is reabsorbed due to aldosterone, but then you can see this little note here that says that chloride follows. Okay, so chloride does tend to follow sodium when we reabsorb sodium, because if we reabsorb a sodium positive and then we also reabsorb a negative, that balances each other out. Okay, so that tends to happen. Now, if you are experiencing though acidosis, and I think this is already in your lecture notes, yeah. So the reason why we reabsorb chloride with sodium is because that's the most common negative ion we have. So we have lots of chloride available. So when we reabsorb sodium, we also reabsorb the chloride, so they balance each other out. However, if you are experiencing acidosis and your pH is too low, then your body has a way to, instead of bringing in a chloride with that sodium, it brings in a bicarbonate instead. Okay? Because bicarbonate is a buffer. So if you're in a place where your pH is too low, instead of bringing in calcium with that sodium, we bring in bicarbonate instead. And that's going to balance our pH as well as balance that positive negative charge. Okay? And that's it. Any questions? We'll talk more about that bicarbonate in part 2. Okay? So I'll see you on lab. Lab will have your quiz first on male, female, reproductive. And then we'll get into the second part.